Well, good afternoon, everyone, and a very big welcome to our first SVI Food Matters webinar. It's unusual circumstances this year, but we hope you enjoy our webinar on osteoporosis. But before we get into things, I would like to acknowledge the traditional custodians of the various lands on which we find ourselves today and pay my respects to the elders past, present and emerging. I extend that respect to any Aboriginal or Torres Strait Islander peoples participating in this webinar today. Well, my name is Karen Inge. I'm chair of the SVI Foundation, and it gives me, on behalf of SVI, enormous pleasure to welcome so many of you today. Many of you would have attended our Food Matters series in the past, and it's wonderful to see that we have some new participants today too. So today's program, it's our first one, it is the first one of three, is going to be on osteoporosis, as you know. And it's interesting because it's a very important topic to many of us, and we have no one better to present than SVI's Professor Natalie Sims. So we're going to start off with that, her presentation, and then I'm going to talk a little bit about the role of food, so why food matters with osteoporosis. But there's a couple of housekeeping things I'd like to go through. After the presentations, we are going to have a short Q&A where you'll have the opportunity to write your questions to Natalie or myself, and there is a Q&A button on the bar at the bottom of your screen. So please feel free to type your questions throughout the webinar. You don't have to wait till the end. You can just type them and we will get through as many as possible. We will only be using the Q&A feature today rather than the chat or the raise your hand. So without further ado, I'd really like to introduce Professor Natalie Sims. Now, Natalie is the head of our Bone Cell Biology and Disease Unit at SVI. She's a professor at the University of Melbourne's Department of Medicine at St Vincent's Hospital and is the Deputy Director of St Vincent's Institute. And she's held that position for the last 2 years. Since starting her PhD at the University of Adelaide in 1990, Natalie has worked to discover new treatments for common skeletal conditions such as osteoporosis and arthritis, as well as rarer disorders like osteogenesis imperfecta that affect the way the skeleton is formed. And after her PhD, Natalie worked at the Garvan Institute in Sydney and then trained at Yale University School of Medicine in the US before returning to Australia in 2001. She is amazing, and as most of the scientists that we have at SVI, she is unbelievably humble about all her achievements. But I haven't got time to really go through them all, but she is quite remarkable. She's an internationally recognised leader in bone research. She's a world researcher, and her team is currently investigating the way that cells within the skeleton communicate with each other to produce bone of optimal shape and strength. So it is with great pleasure that I introduce Natalie to present about knowing your bones, osteoporosis. So thank you, Natalie.
Thank you, Karen. Great. Well, thank you all for joining us. And what I'm going to do today is to talk about osteoporosis, how it happens, why it's so common. And then in the latter half of my talk, I'm going to talk about the work that we're doing within our team at SVI to try and prevent osteoporosis in the future and how we can treat it in the future. So, I just have to work out how to get my slides to move on. So osteoporosis, it's a really major public health problem. And as Karen mentioned, we work on a number of different diseases of the skeleton, but today we're focusing on osteoporosis because it is so common. And in people over the age of 50 in Australia, 1 in 2 women and 1 in 3 men will suffer from an osteoporotic fracture. Now, what do we mean when we talk about an osteoporotic fracture? It's a broken bone, but it's not just any broken bone. It's not one that happens from a car accident or playing football, for example. It's something that happens just from tripping over. tripping over a slippery piece of pavement like this woman has, or a cord within the home, and then breaking a bone. So usually for most people, the first sign that they have osteoporosis is when they break a bone from a standing fall like this. And sometimes they're called fragility fractures. You'll hear that term as well. So these are extremely common. In Australia at the moment, every 8 minutes, someone's admitted to an Australian hospital with an osteoporotic fracture. So for the patient, this is terrible, but it's also a really major cost to the public healthcare system, because if you have a fracture, particularly a hip fracture, it's very expensive to be fixed. And of course, it has lasting effects on your health for the following years. So why does this happen? Why are older people more likely to get osteoporosis? And what I'm going to do throughout this talk is really focus on what happens to the skeleton throughout life, because it's constantly changing. And if you only take away one thing away from my talk, I really want you to understand that the skeleton is always changing. So we know that the skeleton changes from birth until adulthood because it gets much bigger. A baby skeleton is a lot smaller than an adult skeleton, so of course it has to grow. As we get older, and then when we're adults, we reach what we usually refer to as peak bone mass. So that's when your bone is the strongest it's going to be when you're a young adult and when you're healthy, until about the age of 35 to 40, and then gradually the skeleton changes again and it becomes less strong. Even though our height doesn't reduce for quite a long time until we're much more elderly, the bone within the bone gradually decreases and bone becomes less strong. So if we were to draw this as a graph, and we draw bone mass going this way and age across the bottom of the graph, we would see that bone mass, the total amount of bone in our skeleton, increases until we're adults. It's stable for a while we're young and healthy, and then gradually there is this slow decline in the amount of bone that we have in our skeleton as we age. So that's the graph there without the people on it. And then, of course, as we get older, eventually we'll reach a point where the amount of bone that we have in our skeleton is weak enough that it falls below what we tend to call the fracture threshold. And at that point, if you fall over, you're much more likely to break a bone. So it's both bone loss, but it's also increased falls that make older people more likely to fracture. Loss of muscle mass and less physical activity, less physical strength also means older people are more likely to fall when they trip on something than younger people. My focus is on the skeleton, and that's what I'm going to talk to you about now. So there are many, many different influences on bone mass. Why does it go up and then gradually decrease over time? And why do some people have a higher peak bone mass than other people do? Those are the types of questions that we're trying to answer. So there's natural variation in the amount of bone mass that people have throughout life. Some people start with more bone, reach a higher bone mass. Some people start with less bone and reach a lower bone mass. And over time, as people get old, some people lose bone more rapidly and some people lose bone more slowly. And that's because bone is influenced by so many different things. We'll talk a bit about diet, but there's also other things that are very important for both the increase in the amount of bone and the reduction in the amount of bone as we age. So we know that genetics play a role. In the skeleton, there are very, very many genes that can affect the structure of the skeleton. So we're never going to come across one gene that causes osteoporosis because there are so many that are involved. There's also epigenetics. You will have heard talk about epigenetics in terms of many different conditions. So these are ways that your gene expression is changed due to influences that have happened before you're born, and they can also affect the skeleton. Exercise affects the skeleton both as it's forming and as we're aging. Exercise, weight-bearing exercise, makes the skeleton stronger. And nutrition has effects on the skeleton. And this is important throughout life, both in children, having high amounts of calcium to increase the amount of bone as they grow, but also in adults, maintaining the skeleton that we've got. It's really important to have good nutrition. And chronic illness has a major effect on the skeleton, both in terms of the illness itself and sometimes the treatments that people are on. For example, long-term glucocorticoid use over life has a detrimental effect on the skeleton. So there are very many reasons why somebody is more likely to get osteoporosis when they're older than another person. It could be genetics, it could be epigenetics, it could be illnesses they've had through their lives, as well as nutrition and exercise. And menopause is another thing. This is the reason why women get fractures more than men and why they're more susceptible to osteoporosis. Estrogen and progesterone maintain the structure of the skeleton, and when women go through menopause, they do suffer a loss of bone mass, as well as the age-related loss of bone that happens, and that's why it's more common in women. Although, as I showed at the start, 1 in 3 men over 50 is still very common in men as well. Now, the reason why so many things can influence the skeleton and can have an influence throughout your life is that your skeleton is constantly being renewed. It's always changing. And just like your body replaces its skin, and we're well aware of that, we lose skin and then we generate new skin. The same thing happens in the skeleton, but we can't see that happening. And the way that it happens is these cells, and this is what we study in my lab. We study 3 different cell types, and I'll talk about the first 2 and then the other one later. There are cells called osteoclasts, and they dig away at the bone. They remove small portions of damaged bone. So we're talking about microscopic parts of bone. The osteoclasts remove those, and then this cell type comes in, osteoblasts, which then form new bone to replace the bone that's been lost. And this happens constantly throughout your skeleton. Osteoclasts are chewing away at bits of bone, removing parts that are damaged, and then osteoblasts are replacing that so that you've got a healthy skeleton. And this is why your skeleton constantly changes through life. So the reason why bone mass increases and decreases is because of the difference in balance between these 2 different cell types. During the period of time where your bone is building from birth until peak bone mass, the balance is in favour of bone formation. Those osteoblasts are more active, forming lots of bone. There's more building happening, so the skeleton becomes larger and becomes stronger. Between early adulthood and old age, the balance shifts in the other direction. Those osteoclasts are more active compared to the osteoblasts. So there's more digging happening and less filling or not enough filling happening to fill each of those microscopic holes through the skeleton. So over a long period of time, that tiny imbalance in those microscopic areas throughout your skeleton gradually causes loss of bone. And that's why our bones become weaker and have less strength as we become older. So how can we prevent osteoporosis? The simplest thing to do, of course, is to slow down bone loss. It's very hard to control peak bone mass because all of these factors influence as we get older, as we grow. But what we'd like to do is to slow down bone loss so that it doesn't reach that fracture threshold. And the way to do that, most common treatments that are used for osteoporosis, and possibly some of you on the call are taking these treatments, are inhibitors of digging. So these are treatments like bisphosphonates or teriparatide, Prolia or Actimel, denosumab. There's lots of different names for these. What they do is they inhibit the activity of those digging cells so that there's less resorption and less destruction of the skeleton happening. What this means is it can stop the bone loss from continuing further. So in this example, I've drawn a purple line from the bottom. So it's a person who has a lower level of peak bone mass. They haven't yet reached the fracture threshold, but they know they've got low bone mineral density. So their doctor has suggested they go on the treatment, and that keeps their bone mass above the fracture threshold. For someone else who might have had a fracture already, they can then be put on this treatment and their bone loss won't get worse. What it can't do is to actually replace the bone and bring that patient up above the fracture threshold. So it won't give them a healthy skeleton. And that's what we would like to be able to do in the future, is to give people a healthy skeleton. One of the reasons why these treatments can't put bone back and make bone increase further is because these 2 cell types communicate with each other. And that's important. That's why our bone is normally so healthy, because the activities of the 2 cell types are balanced. When we inhibit osteoclasts with the treatments that we've got, over time, it eventually inhibits the activity of osteoblasts as well, because normally we want those activities to be matched. So what we need to be able to do is to get over that inhibition. So what we're doing in our lab, and I'd like to focus on that now, is to try and work out other ways that we can improve bone health, is to move away from thinking only about peak bone mass. So this is what I've talked about so far, the amount of bone that's in our skeleton. What we're starting to think about more and more is peak bone strength, because we're learning that the strength of bone is not completely determined by how much bone is in the skeleton. And I'll talk to you about this now. So we know that patients who have low bone mineral density when they've had a scan, so you may have had a bone scan, or sometimes called a DEXA scan, to know what your bone mass is like. If you have a low bone density, you are more likely to fracture. But most patients who fracture, 60% of patients who have fractures just from a standing height, actually have a normal bone density scan. So this means there are other things that contribute to fracture other than somebody's bone density. So what are some of those things? Bone material quality can also affect the strength of bone. The distribution of bone in the skeleton, you want to have bone in those places where you're most likely to fall. And one of the things about bone distribution is there's actually two types of bone in our skeleton. So this is an X-ray, and in this I'm showing you an ankle because you can see two different types of bone here, and you can see how terrible fractures can be. There's two types of bone. One of them is cortical bone, which you can see up here. That's the thick outer shell. So if you feel your wrist, what you're feeling there is cortical bone. That's the outer shell of your skeleton. But in other parts of the skeleton, trabecular bone is most important for strength. So you can see the very, very fine lines in the heel bone here, and they're trabecular bone. So while this part of the skeleton doesn't have a thick cortical shell, what it's made up of is a very fine honeycomb-like structure that provides the strength for that part of the bone. And we know that the treatments that we have so far are very good at treating trabecular bone, at reducing bone loss that happens in the vertebrae, which are mostly made up of trabecular bone, but they don't perform quite so well at preventing hip and wrist fractures. So that's shown here, much less reduction. And this is with the new therapy that's just been approved in Australia, romosozumab. So it's very good at reducing fractures in the vertebrae, not so great at reducing fractures in the hips and the wrist, but it's the best we've got so far. So why is that? The reason for that is that vertebral bone are kind of like this heel bone. They don't have a thick cortical structure. Their strength comes from the honeycomb network inside the bone, from that trabecular bone. Whereas at the hip and the wrist, their strength comes from the cortex. So about 3/4 of the strength of the strength in the wrist and the hip comes from the cortical bone. And that's because these are bones that are a little bit more like this. They're called long bones, and the cortical bone contributes to the strength. So when somebody breaks a hip or a wrist or a leg in this case, the fracture happens through the cortex, whereas in the vertebrae, it happens through trabecular bone. What we're doing is to try and understand what makes cortical bone strong, because the field has focused for a very long time on trabecular bone, but there's more we need to know about the cortex. When we think about cortical bone, the easiest analogy to think of is really to think about pipes and tubing. Think about your plumbing. We know that different pipes have different strengths, and that could be because they're made of different materials, they have different widths, the material has different thicknesses. It's exactly the same with our bones. So if we were to take a cross-section of a femur, we would see that obviously the one that's thicker is going to be stronger, the one that's wider is going to be stronger, and the one that's made of a better quality material is going to be stronger. So the work that we're doing is to try and understand what are the things that determine the material strength of bone and how bone is organized around the central axis that makes pipes, bones of the same size, but that are stronger than what they were before. So first I'll focus on bone material and some work that we've been doing on that. So we all know that bone has got calcium in it, it's got mineral crystals, it's hard, but bone actually has two really important components. One of them is collagen. You know that you've got collagen in your skin. There's many, many types of collagen throughout the body. And bone has a specialized type of collagen called collagen type 1, and it forms a helix, kind of like DNA, but it's got 3 molecules in a helix, and it's very flexible. So that's what makes bone flexible. When you fall and your skeleton is healthy, you want it to be able to bend a little bit, not just to be made of mineral. And our skeleton also, of course, has mineral crystals. And this is from a fairly recent paper that used some really, really high resolution imaging. This is not our data, but they're showing the collagen helix in the middle here, shown by the tube. And then surrounding that are these red mineral crystals that form around it and make it hard. So bone has to be both flexible and hard. And it's the balance between flexibility and hardness that we are interested in. And we're also interested in these cells. These are my favorite cells. They actually sit right inside the bone, in between those collagen fibers and the mineral crystals, and they're responsible for maintaining the hardness of the tissue. You can see that these cells have got long projections that reach out and touch each other, and it's through these projections that they're able to send signals within the bone. The cells sit within little fluid-filled canals and pockets inside our skeleton, and they're tiny, and they can send signals to and from each other, but also to those osteoblasts, the bone building cells, and the osteoclasts, the bone digging cells that I introduced you to before. They form a very complex network, and I like to think about the network as being very similar to the brain. So within your skeleton, you've got around about 42 billion of these cells forming 23 trillion connections. So it's not dissimilar to the number of neurons that you have in your brain communicating and telling you how to think and what to do every day. Our brain is also communicating within itself in a similar way. Sorry, our skeleton, our bones, are communicating in a similar way to the brain. So we've been studying these cells, and we have now shown that osteocytes within the skeleton control bone strength actually from the inside of the skeleton, not only by controlling bone mass, but also by controlling composition. We've used a mouse model that has bones that are very brittle. So mice are quite useful because we can look at their bones and actually see how strong they are by testing their strength. We saw with mice that had a protein called efferin B2 missing, specifically in osteocytes, that they had bones that had normal mass, normal size and normal dimensions, and a normal density scan. But when we tested their strength, they broke very easily. To understand what had changed in these bones, we couldn't use any normal tests. So we went to the Australian synchrotron in Melbourne, and we analysed the internal structure of their bones. What we found by looking at very thin sections of the bones of these mice and a tiny region of the bone and shining bright infrared light through it was that these bones had differently arranged collagen to normal. It was more squashed, and they had a higher level of mineral than normal. So we're now studying this pathway to work out how we can manipulate this pathway to increase bone strength, specifically by changing the material of the bone. Because if we can understand this treatment, we could develop new treatments that can strengthen the bone that's already there to make the material stronger. The other approach that we've been taking is looking at how osteocytes control the organisation of the bone. So I talked to you about how there's both cortical bone and there is trabecular bone. As we become older, our cortical bone becomes more porous. It actually becomes more like trabecular bone. So these are some very high resolution scans showing you the cortical bone in someone as they age. It's not the same person. They're just different bones taken from people at different ages. And you can see that even though there is cortical bone, the older person, the 90-year-old, much of their cortical bone has got very large holes in it, and it looks more like that honeycomb network I was talking to you about before. So we've been studying how this process happens, and our most recent work has been studying a protein called GP130 and how that controls the holes within the cortical bone. So this is a different way of looking at those osteocytes here on the left And you can see they're organized in nice rows, and they've got their projections going out and sending messages to each other. And the collagen is arranged in layers. You can kind of see it looks a bit like a layer cake with very thin lines going through it. Layers of collagen are very strong. And in the cortical bone in this particular sample, it's a young bone sample, there are very few holes, but they're needed for blood vessels to get through. When we changed the expression of the protein GP130 and made it hyperactive, what we saw is that even though there's more bone, it's weaker because the collagen is less organized, the cells are less organized, and there are a lot more holes within the bone. So we're using this model to try and understand how these holes form and how we can refill them, so that in someone who is older, if they have a skeleton that is really porous, we would be able to fill those holes and make the bone stronger. So if we go back to our original graph that we were talking about before, what our hope is that in the future we can make bones stronger by stopping the formation of holes within the cortex so that people don't reach the fracture threshold. And in those patients that have reached that threshold, we can increase the mineral content and we can fill the holes that are there. So thank you very much, and I'll hand back over to Karen.
Well, thank you very much, Natalie. It's fascinating. You know, I think that we've all learned a lot, and I think so many of us think that bone is just a simple structure, but it's, I think you've certainly highlighted in a very short period of time that there's a lot more to bone than that. So I would like to now flesh out a little bit of what Natalie referred to in terms of why diet is important. So we'll move on to the next part of the presentation, the webinar. So this is just really a little snapshot of why food matters and for bone health. So a lot of this information you would already be very aware of, and what foods are very important for increasing or helping to look after your bones. And the nutrient that I think everybody would be aware of is calcium. And the richest source of calcium in our diet, or the most available source, actually comes from dairy foods. And calcium is really important all the way through life, because calcium is not only important for bone formation and maintaining bone strength, but it's also important for nerve transmission, for teeth, for muscle contraction. So if we're not getting enough calcium in our diet, then the body will take from, our bodies will take from the reserves. And as you know, calcium is stored in the bones and teeth. So I think getting back to that. The other nutrients that are also very important in maintaining bones and for healthy bones, obviously vitamin D, which is known as the sunshine vitamin. And from a diet perspective, vitamin D is very challenging to get from food sources. You're probably aware of fatty fish and full-fat dairy products that will provide vitamin D, which is a fat-soluble vitamin. But what you mightn't be aware of is that mushrooms, when left out in the sun, in the ultraviolet light, will actually be a significant source of vitamin D. And if you slice them up and leave them out in the sun for about 5 minutes, you will get an increased amount of vitamin D. So I could just see everybody out there with slicing their mushrooms. But remember to bare your skin at the same time, and because that's really where most of the vitamin D is manufactured. On the skin. So that's important. Vitamin D is very important to help calcium to be absorbed. And the other nutrients that are also important are vitamin K and magnesium. Magnesium is spread throughout the diet through nuts and seeds and a lot of plant foods. And vitamin K is a fat-soluble vitamin, which is also found in a lot of green leafy vegetables, for example. So that is, again, very important to get a myriad of foods. But how much do you need of these foods? Well, with dairy, around 3 to 4 serves of dairy foods every day. For people, for women post-menopause and for men over 70, that's the requirement. And that's, for a lot of people, that's a lot of dairy. And if you can't tolerate dairy, or you have a lactose intolerance, you might want to, or you're vegan, for example, you might want to go to some plant sources. And many of the plant-based milks that are available today, like oat milk and rice milk and soy, are fortified with calcium. So they are good alternatives besides other foods like nuts and seeds and sardines. And of course, it is also very important to get all the other nutrients that we spoke about from eating at least 2 serves of fruit and 5 serves of vegetables every day. And remember to eat the rainbow. Lots and lots of colour will give you more variety. I just wanted to cover a couple of things that you might not be aware of, but there are certain components of foods which can actually enhance the absorption of calcium and also interfere with the absorption of calcium or increase the calcium losses through the body. But these are the positive aspects. Tea, you might not realise, but tea actually is very high in polyphenols, and there have been studies to say that drinking tea can actually decrease fracture risk. Now, it's early days and we don't really know how much tea you do need to consume, but tea is thought to be a good beverage. It shouldn't replace all the milk in your diet, but it's certainly beneficial. When we're looking at green leafy vegetables, which I mentioned before are a source of vitamin K, you do need to be mindful that some of them are very high in oxalates, and oxalates will interfere with the absorption of calcium. So although they are a source of calcium, we've heard about broccoli, etc., and vitamin K, they can interfere with the absorption of calcium. So the only vegetables, green vegetables, that seem to not be high in oxalates and not operate this way are the Asian greens. So your bok choy and Chinese cabbage are thought to be good sources of calcium and fairly well absorbed. Now, with all of the legumes and lentils, etc., they're high in phytates, and phytates can also interfere with the absorption of calcium. So it's a good idea to soak these legumes and lentils overnight and then pour off the water, and they'll be easier to absorb and not have the phytates will be released, and it won't have a negative effect on the absorption of calcium. You've heard of activating almonds. Well, there is a bit of a truth in that. Sprouting them, soaking the almonds overnight reduces the phytic acid content. The other aspects in terms of how to keep calcium in your body, these are the dietary factors that we know. We know that a diet that's very high in salt, so an excessive amount of salt, will cause calcium to be excreted from the body. So you will lose calcium. So it is important to be mindful of that and just be mindful of not having a lot of Asian sauces and a lot of extra salt in your diet and salty foods and a lot of fast foods, because they can interfere with the absorption of calcium and could cause calcium, well, not so much the absorption, but cause calcium to be leached from the body. And there's been a lot of discussion about soda water and bubbly drinks and phosphoric acid in these drinks. Well, it seems to be that soda water and aerated waters are absolutely fine as far as not increasing fracture risk, but it appears that cola-containing drinks are not healthy. And some work's been done with young people, and we're not really sure of whether it's the caffeine in these drinks or whether it's a combination of the phosphates and the caffeine, or whether it's just purely a matter of young people drinking these drinks and not having enough of the calcium-rich drinks like milk in their diet. And the only other factor that I wanted to talk about was the impact of alcohol. And fortunately, or unfortunately, a lot of the work has been done with alcoholics, and we know that excessive drinking is not good for bone health. It's certainly deleterious for bone health, but there haven't been really many studies done in terms of looking at low to moderate drinking. So I think safe drinking, which is around no more than 10 drinks a week with the new draft guidelines doesn't seem to have a negative effect on bone. So, and just my last slide is really the big debate, food versus supplement. Well, food first is always very important, because when we get our nutrients from food, we're getting all the components that work synergistically to help absorb the key nutrients from food. But if nutrients are not able to be met through food alone, then supplements may be appropriate. So I'll leave it there. It's a bit of a snapshot, and we'll move now to the rest of our presentation. And obviously, I'd like to thank Natalie very much for her presentation. And it's wonderful to see, Natalie, that we do have quite a few questions. So I thought we would tackle some of those now. So the first question to you, Natalie, is there any evidence to suggest significant differences according to population groups, for example, in the Japanese?
Well, absolutely. Yeah. So as I mentioned, genetics play a really important role in determining the strength of the skeleton. And different populations we know have got differences not only in bone mineral density, but also in the size and differences in the shape of their bone as well. And that can affect bone strength.
Thank you. And then we have another question, which is, if you're older than 50, is bone loss homogeneous over the body, or is it more focused in certain areas like legs, arms, hips?
Actually, that's a really great question, and I don't think we know the answer to it. We do know that bone loss happens throughout the body, but we don't know if it affects some parts of the body more than others. And it's a possibility that it could be different in different parts of the body. Usually when bone density is assessed in people, it's assessed in the spine and in the hip, because that's where fractures happen, and that's what we know the most about. But we don't know about other areas at all.
Mm-hmm. Is there a cut-off percentage for bone density at which medical treatment or supplements are required or desirable?
Yeah, the decision about whether someone should be treated with the current therapies that are available is based partly on bone density, but it's also based on whether the person has had a fracture already. So if someone has had a fracture already, we know that they have osteoporosis and that they will need treatment. So it's a combination of both of those. And when people are looking at bone density, it's not the number itself, it's how that number compares to the general population at that age that's used as a cutoff. So talk to your doctor, but talk about both of those things, bone density and whether you've had a fracture.
Are there currently any medication or supplements that can help build bone that you could comment on?
A terrific question. Yes, there are. So Forteo is a treatment that can build bone mass. Unfortunately, it can only be used for a short period of time. So it can be used for 18 months, so to give someone a boost in their bone mass, and then usually they're put on an anti-resorptive, those treatments that I talked about earlier. The reason why people can't be treated for longer than that is we don't know how safe it is for someone to be on one of those treatments for a long period of time. So the current law is that people cannot be treated for longer with those until we know more.
Just one more question. We have so many questions, we won't be able to get to them all. But with regard to Prolia, at what age should one use it from, and for how long can it be taken?
Yeah, so Prolia is one of those osteoclast inhibitors. It seems it can be taken quite safely for, we know, at least 10 years. That's what the studies have gone out to, and possibly longer. And again, whether someone should be treated with Prolia, it depends both on their bone mineral density and on whether they've had a fracture or not. So, yeah.
Well, thank you, Natalie. Look, there are so many more questions, but I'd just like to assure people that we will answer your questions. Natalie has made a promise, and there's a couple for me, that we will get back to you and answer your questions within a week or so. Yep. So that really brings us towards the end of our webinar. So I'd like to thank you very much for coming. But I'd like to also let you know that we have 2 more webinars coming up on the Food Matters series. The next one will be on Wednesday, the 19th of August, with the wonderful Dr. Ramin Cheyenne, who will talk about cancer and its connection with nutrition, which should be absolutely fascinating. And our final webinar will be on Thursday, the 17th of September, which will be from our SBI Director, Professor Tom Kay. And of course, as you know, he will discuss a lot of the work that we're doing at SVI to better understand type 1 diabetes. So both seminars should be very interesting. But if you would like any further information on the webinar series, or you're interested in hearing more about the work that Natalie does, or you'd like to support the work that Natalie does, or you'd like to find out about anything else that we're doing at SVI, please email Nicola Petgal or Kate Barnett at the foundation, and it's foundation@svi.edu.au, and I'm sure that they'll be able to help you. So that's it from us. We'd like to, Natalie, thank you so much for today, and thank you to all our participants, all the friends of SVI, for joining us. It's marvellous to have so many of you, and keep safe in this unbelievable environment we find ourselves in. We look forward to seeing you at